In the previous video, we have been discussing about glycolysis, in which one glucose molecule it gets converted to two pyruvate molecules along with the generation of two ATP molecules and in a 10-step reaction. So we also understood about various pathways which are related not only to glucose but to other different carbohydrates and how it is being metabolized via glycolysis. So in this video we will be learning about tricarboxylic acid cycle, which is also called as Krebs cycle. So this Krebs cycle, this was discovered by Hans Krebs in 1937 and he was awarded Nobel Prize for Physiology in the year 1953. So this is a pathway in which this pyruvate, which is formed in the earlier glycolysis, it should be further oxidized to carbon dioxide and water in aerobic condition. Okay, so this is called a cellular respiration, and this takes place in mitochondria because all the enzymes which are required for this metabolic pathway is present only in the mitochondria. And the biomolecules like glucose, amino acids and fatty acid, it serves as a fuel or the driving source for this particular pathway to take place. And this is going to serve as a hub of metabolism. The intermediates which are formed in this particular pathway, it is being siphoned to various other pathways as well to form the precursor for many amino acids and also fatty acids. Okay, so here whatever intermediates which are being used up, it has to be regenerated or it is being regenerated, and this is called as anaplerotic reactions. Similarly, this is also going to be both anabolic and catabolic in nature, because it whatever biomolecule which is entering into this pathway, it will be broken down to H2O and carbon dioxide. Similarly, the intermediates which are used up formed in this pathway, it is being siphoned or it is being used as a building block for some other metabolic pathway. Okay, so that's why it is also called as amphibolic pathway, and this is a closed loop of reactions. Whatever oxaloacetate which is being consumed during this pathway, it will be regenerated as well. Okay, so hence it forms a closed loop of reactions, and this reaction is going to be a 10-step process, and this is going to be a very important pathway because of its because it serves as a hub for various metabolic activities. Okay, so here this explains the amphibolic nature of this particular pathway. So here we can see that glucose, okay, which is getting converted to pyruvate via glycolysis, and similarly we have the other fatty acids and amino acids as well. So all these things, it enters this TCA cycle, and so this is going to be the catabolic in nature because it is going to be broken down. And here whatever intermediate that is formed during this TCA cycle, it is being utilized for various other pathways. Okay, so for fatty acid synthesis or cholesterol synthesis or as for production of ketone bodies, all this acetyl-CoA is being utilized as an intermediate or as a building block. So this is going to be anabolic in nature. Okay, so this acetyl-CoA or this pathway, it serves as amphibolic thing. Okay, and here if you see in this diagram, we can see the same thing. Okay, so here as a result of the citric acid cycle, you have the production of two carbon dioxide molecules. So again, this explains the role of this TCA cycle in the various other metabolic pathways. Okay, it plays a vital role in fatty acid synthesis, in amino acid synthesis, purines, and porphyrin synthesis as well. Okay, so what actually happens here is this pyruvate, okay, which is formed during the glycolysis reaction, it is being oxidized to yield a 2-carbon compound, and that is called as acetyl-CoA. And this acetyl-CoA, it enters the TCA cycle, and this is a strictly aerobic process in which 12 ATP molecules are produced when this TCA cycle is coupled with oxidative phosphorylases. Okay, So in the TCA cycle, we don't have much of energy or ATP generation, but when this particular reaction is being coupled along with the oxidative phosphorylation, it results in the production of around 12 ATP molecules. And this TCA here it is called the citric acid cycle because all the first three intermediates which are produced during this course of this reaction, it possesses three carboxylic groups. Okay, so hence it is called as a citric acid cycle, and this is going to be a metabolic pathway with 8 different steps, and here we can see redox and dehydration, hydration and decarboxylation. These are all the 4 steps which is going to happen repeatedly, whereas in the previous glycolysis we saw that phosphorylation and dephosphorylation was happening predominantly. And here as a result it produces around 1 ATP or GTP is produced along with 3 NADH molecule and 1 FADH2 molecule. Okay, so let's see about the various pathways now. Sorry, different steps that is taking place during this pathway. So the first step, which is the prerequisite for this TCA cycle, is the production or the synthesis of acetyl-CoA. So here this acetyl-CoA is produced from pyruvate, and pyruvate is obtained from the glycolytic pathway. Okay, so this reaction is being catalyzed by the enzyme pyruvate dehydrogenase, and this is the most important enzyme for this particular step to take place. So here we can see that this is an irreversible reaction, and here acetyl-CoA conversion, it takes place along with the evolution of CO2 group, that is carbon dioxide, and this enzyme is very unique because it is a multi-enzyme complex. 3 subunits like E1, E2 and E3, they together it forms the entire enzyme, and these are all its corresponding subunits. Okay, and also we can see that for the activation of this particular enzyme we need some cofactors. Okay, so there are 5 different cofactors which are essential for the activation of this enzyme. Okay, thymine, pyrophosphate, FAD, CoAsh, NAD and lipoate. And this is an important step because this forms the connecting link between glycolysis and TCA cycle. So we already know glycolysis, it takes place in the cytosol. Okay, and this is the final product of glycolysis, that is the pyruvate. And this TCA cycle, it takes place in the mitochondria. So this pyruvate is being transported via this transporter protein. Okay, and 3 reactions take place here. The first one is a decarboxylation reaction. Okay, so we see this pyruvate is getting oxidized, right? So this CO2 is being removed, and we also see that there is a electron acceptor here. Okay, and NAD plus acts as the electron acceptor. So for this NAD plus we need a hydrogen, right? So that is obtained from this coenzyme A. Okay, so for this from pyruvate we need the formation of acetyl-CoA. So this CoA group, it's obtained from this coenzyme A. So coenzyme A is nothing but CoAsh. Okay, CO it's CoA, which has a sulfur and a hydrogen group. Okay, so from this hydrogen only, this NADH, it obtains its electron and it becomes NADH and H plus. Okay, and so finally we have acetyl-CoA molecule CH3, C double bond O and SCOA. Okay, so S is obtained from this sulfur group of CoAsh, and CoA is this. Okay, so this is what is the first step in TCA cycle. Okay, so the same thing is explained here. This is a 3-step process. Okay, so next, whatever acetyl-CoA it is formed, okay, it is going to enter the TCA cycle, and we can see these are the different steps. Okay, so first acetyl-CoA, it gets converted to citrate. Okay, this is catalyzed by the enzyme citrate synthase, and then you have a series of water is being removed and then again water is being added. Okay, and this is catalyzed by the enzyme aconitase, and this results in the production of isocitrate. Okay, and this isocitrate is further reduced by isocitrate dehydrogenase, and that results in the production of oxalosuccinate. Okay, so here again, whatever hydrogen, remember one thing, whenever there is a dehydrogenase enzyme, it results in the production of a or removal of a hydrogen ion. Okay, so that hydrogen, it will be accepted by a electron acceptor, which could be either NAD plus or it could be either FAD plus here in this case. Okay, so here the hydrogen which is removed here, which will be accepted by NAD plus, and finally again one more decarboxylation step takes place here. Okay, and that results in the production of alpha ketoglutarate. Okay, so this alpha ketoglutarate is an important intermediate for the synthesis of the amino acid glutamate. Okay, and this alpha ketoglutarate, it can be further reduced and it forms succinyl CoA, and this succinyl CoA, it can be reduced again, it can be acted by succinate thiokinase, and this results in the production of succinate. And meanwhile here we have the production of a GTP molecule. Okay, and this succinate is again reduced to form fumarate, and here again this hydrogen electron is being accepted by FAD. Okay, and fumarate, it gets converted to malate and by the enzyme fumarase, and malate, it's further reduced to oxaloacetate. Okay, so the oxaloacetate which was consumed in the first step, it's again regenerated here. So now we'll see the step-by-step process. Okay, so you can see this pyruvate, right? Pyruvate is getting converted to acetyl CoA. Okay, and we see this is pyruvate CH3, C double bond O, COO minus, and we have the CoAsh, that is a coenzyme coming into the scene. So this CoA, it gets it has to be added to the pyruvate as such, and the first step is the removal of this CO2 and addition of the COA in place of this CO2. Okay, and this hydrogen, it is also going to be used for the reduction of NAD plus. Okay, so finally you have acetyl CoA. So this acetyl CoA, it is going to undergo a condensation reaction with oxaloacetate. Okay, so this happens in presence of water, with the addition of water. So we can see that this one remains as such. Okay, so just because this is a condensation reaction, right? So here there is some rearrangement taking place here in this carbon position. Okay, so here we can see that to this carbon position, this is going to be reduced first. Okay, there is a reduction reaction happening here, and then the COO gets rearranged and it's coming to this side. Okay, so that's what you see here. Okay, you have a OH here and the COO minus, and now to this position, this to this third carbon, we have the addition of the pyruvate group. Okay, so, acetyl group. Okay, so this CH3 COO, it has to be attached. Okay, so it finally forms CH2 COO, because one hydrogen from this, it has been added to this one, to this O group of oxaloacetate. Okay, so finally you have the formation of the citrate, and the citrate, it has 3 carboxyl groups. Okay, 1, 2 and 3. Okay, and the next step is going to be catalyzed by aconitase, and the first one, here you have the removal of water. So we can see for water means this OH group and then this hydrogen. Okay, this is being removed, and next this is an unstable compound. So next again the same, this is called cis aconitate, and again one more molecule of water will be added, and the same enzyme is going to catalyze the next step. Okay, so here what happens, you have addition of water, right? So here we have this addition of water, and here it remains as such, and here again. Okay, so H and OH, and Now this compound, it is called as isocitrate. So this again has 3 number of carboxyl groups. And next step is isocitrate dehydrogenase. So I have already told you, whenever there is a dehydrogenase, there will be removal of H2. Okay, so let's see here. You can see that the OH group which was present here, right? The OH group which is this OH group which is present here, and the hydrogen uh, which is present here. Okay, so this has been cleared. So this is one hydrogen and this is one hydrogen. It has been removed now, and that is replaced by a double bond. Okay, so here you have a double bond, and so one hydrogen is used for the reduction of this NAD plus to NADH, and the other hydrogen, it dissolves in the medium. Okay, so that is how this NAD plus works. And next you have the isocitrate dehydrogenase. Okay, so this compound is called oxalosuccinate, and this is again an unstable one. So this is again being reduced by isocitrate dehydrogenase. So here we have the production of carbon dioxide or evolution of carbon dioxide, and we can see that the COO and CH2 is there, and the hydrogen which was present in this position, it is being removed. Okay, so if you can see, okay, this hydrogen is removed and this carbon dioxide is also removed here. Okay, so you have C double bond O and COO minus, and okay, so this compound, this is called succinate. Okay, sorry, alpha keto glutarate, and this is a very important intermediate, and this is going to serve as a building block for an acid like glutamate. Okay, so next step is going to be catalyzed by one more dehydrogenase, that is alpha ketoglutarate dehydrogenase. So here we need a product of succinyl CoA, so which means that there has to be an addition of a CoAsh group. So you have a CoAsh group, and this CoAsh is being removed as CO2. Okay, and since this is a hydrogenase, we need an electron acceptor, that is NAD plus. So here we can see that the hydrogen which is here we have the addition of a CoA group, right, and along with the evolution of CO2, right. So as you see here, this CoA group has been added as such, and this results in the formation of succinyl CoA. And next, this succinyl CoA has to be converted to succinate, and this takes place in the presence of a energy molecule like ATP or GTP. So this is catalyzed by the enzyme called synthetase. So there is a difference between the synthase and synthetase. So synthase, here we have the reaction which is taking place in the absence of ATP, whereas for synthetase, driving source is required, that is energy molecule is required for the reaction to take place. Okay, so here if you can see, the CoA group is removed now. Okay, so it is eliminated as CoAsh. Okay, and this keto group which was present here, this is going to be further oxidized, so it results in the formation of acid. So this acid is called succinate. So you have two acids now. Okay, and in between the two acids you have two alkyl groups. Okay, so that is succinate. And next, this succinate, it's converted to fumarate by the enzyme succinate dehydrogenase. So here again we need a electron acceptor. So here the electron acceptor is FAD. Okay, it's not NAD. Okay, please remember that. And here again, usually NAD gets reduced to NADH plus a H. Okay, so the other H, it dissolves in the medium. Here FAD, it gets reduced to FADH2. Okay, so both the hydrogen, they gets added to this FAD. Okay, and you can remember it very easily. Okay, the product is fumarate, F for fumarate. So similarly, it is being it uses FAD as the electron acceptor. Okay, and then you have the formation of fumarate now. So this fumarate, it's getting converted to malate now. Okay, L-malate. And this enzyme fumarase, it is going to be a very stereospecific enzyme. Okay, so what it does here, this reaction takes place in presence of water, right? So you have addition of hydrogen over here, and also another hydrogen gets added over here, um, right? And sorry, another OH group adds over here. Okay, so H2O means H and OH. So one hydrogen it gets added over here, and here you have the addition of a OH group. Okay, so that results in the formation of L-malate, and this L-malate it's finally getting converted to oxaloacetate by the enzyme malate dehydrogenase. So here the electron acceptor is NAD+. Okay, so whatever oxaloacetate has been consumed, it is being regenerated here, and also we can see that the one molecule CO2 is getting removed, and sorry, two molecules of CO2 is getting removed here. Okay, so this acetyl CoA it also has two carbon. It's a two carbon compound. So whatever carbon that is removed in this step, it is actually the acetyl CoA from the previous cycle. Okay, so the new carbon from the acetyl CoA which is freshly entered into the pathway, it will be removed in the next round of the TCA cycle. Okay, so this process it keeps on continuing. So in the remaining steps, you can see that further description of this pathway in the next video. Okay, thank you.